Hey buddies, how are you all doing? So hope you all are doing great. A very warm welcome buddies to this class. So this is the session which you guys were eagerly waiting for, that is excretory products and their elimination. So this is a one shot of this whole chapter. So let's see about this chapter. This chapter is very interesting and easy as well. So I'll try to give you everything in a short way. So let's get ready. Excretory products and its elimination, that means here we will be talking about the different kind of excretory waste produced by the organisms. Different organisms produce different excretory waste. Like some organism, they excrete ammonia, some excrete urea and uric acid. All these different type of excretory waste, they are important. In fact, when they remain in our body, they are toxic. Clear? So on the basis of toxicity also we divide them. Now next we will be talking about the different group of organism. They are having different excretory structure. For example, in case of vertebrates, what do we find? We find kidneys. So we will be looking at the detailed structure because chapter is a part of your human physiology. So this chapter, you know, it's important for your NEET point of view. So let's not delay and let's start. Excretory products and their elimination. This we will be dealing today. This chapter, very important. 4 to 5 questions you can directly expect. 4 to 5 questions you can directly expect from this chapter. Now this year, you know, the syllabus has been released, and the syllabus which has been released, the digestion and absorption chapter has been deleted as per the syllabus, as per the NEET syllabus. So the weightage of human physiology part has increased. That means the chapter earlier, the 4 to 5 question used to come from this chapter. Now the chances are the number of question will definitely increase because the syllabus has been reduced. Let's talk about the types of excretory waste. As I told you, different group of organisms, they produce different kind of excretory waste. You can see on the screen, we talked about, one second guys, we will be talking about the ammonia, urea and the uric acid. Now, please remember, excretion means removal of the excretory waste. Egestion means removal of the undigested food. Here, ammonia is there, right? Here, there are some organisms who excrete ammonia. These organisms, they are termed as ammonotelic organisms. These are termed as ammonotelic organism. These are termed as ammonotelic organism. And the process of this elimination is termed as ammonotelism. Ammonotelism. This is the process, ammonotelism. This is the ammonotelic organism, ammonia. Now, ammonia, you know, that is a excretory waste produced in our body because of the amino acid. Because of the removal of the amine group, like that is the NH4 positive. So this is eliminated from the body as NH4 positive. Here for the elimination of NH4 positive, kidneys are not responsible. So their major excretion occur through the gills. So they are found in the aquatic amphibians, tadpole of the frog, aquatic insect, bony fishes and mostly invertebrates. Because their rapid removal is important. Sometime from the body surface, sometime depending on the organism, they are removed from the body the moment it is secreted. Because we cannot keep this in our body for a longer time. Are you getting this point? Now, next is urea. Urea is that waste which we produce. So 20 to 25 gram, if your NCERT says 25 to 30 gram of urea, we excrete. How much? 25 to 30 gram of urea we excrete per day, right? Our major nitrogenous waste is urea, but the nitrogenous waste is produced in the form of ammonia. Now this ammonia is converted into urea with the help of ornithine cycle occurring in the liver. Now, organisms which excrete urea are your mammals, frog, toads and the cartilaginous fishes. Bony fishes come under this category, whereas cartilaginous fishes, they come under this category. Here they are involved in the osmoregulation as well. Now, uric acid, that means elimination of the crystals of the waste, which are termed as uric acid. Uric acids are the least toxic. They require minimum amount of water for the excretion. Maximum amount of water is required for ammonia. Moderate amount of water is required for the urea. Minimum is required for the uric acid. Now these organisms, they are termed as uricotelic organisms. uricotelic organism, right? And process is termed as uricotelism. Uricotelism. Clear? Are you getting this point? Whereas urea, this is by the ureotelism or ureotelic organism or ureotelic telism. Process is termed as ureotelism. Clear? Here the organism are birds, reptiles, your land snails, insects and the cockroaches. Different group of organisms, they are having different structures present in their body which helps in the excretion. For example, protonephridia. This is seen, or the flame cell, this is seen in case of the platyhelminthes. In fact, in case of rotifers also, you will find the protonephridia are present. Let's look at the next group of organism, that is the earthworm. In earthworm, you will find the nephridia are present. Different type of nephridia are present in the earthworm. Like septal nephridia are there, like integumentary nephridia are there, the pharyngeal nephridia are present, which helps in the excretion. Now the next one is a Malpighian tubule, which is seen in the case of the cockroaches, that is other insect. Insects, they are having Malpighian tubules for the excretion. This you all have studied in the case of the cockroach. Antennary gland and the green glands, these are found in case of the crustacean. Kidneys are found in majority of invertebrates. I hope these points, they are very much clear. I have only taken those examples which are there in your NCERT. So it is important for your NEET examination. Now, jumping on to the next concept, that is a human excretory system. So, human excretory system, that means this is the one which is involved in two different processes. One process is termed as excretion, and the second one is the osmoregulation. Now, what do you mean by excretion? Excretion means removal of the waste. And the waste in case of the mammal, that is the urea, right? Urea, that's a waste. Next is the osmoregulation. Why am I saying osmoregulation? Because the kidneys are involved in the maintenance of salt and water balance in our body. So how it is important? This is important for the maintenance of salt and water balance in body, or basically in the body fluid. This plays a great role. Here, to talk about the human excretory system, that means various structures are present. The first structure which is present is the kidneys, right? Kidneys are not only structure which is present in the excretory system. In fact, the ureter is also there. In fact, the urinary bladder is also there. And last but not the least, the urethra is also present. Now let's look at the arrangement of these. Now you know these are arranged in the lower abdominal part. If you look at here, you will find the kidneys are present. We have a pair of kidneys. From the kidneys, these ureter comes. Both the ureter, they join to the sac-like structure termed as the urinary bladder. Urinary bladder open to the outside with the help of urethra. Urethra is long in case of male and it is short in case of female. Clear? Now you can see the kidneys are present. Very important question which has been asked from this topic is position of the kidney. So you should know the kidney is present between the last thoracic vertebra to the third lumbar. That is T12. This is written over here. T12. The T12 is what? Thoracic. Last thoracic. That is the last 12th thoracic. To the third lumbar it is present. Here if you look at the kidneys, the left kidney is slightly towards the upper side and the right kidney is slightly towards the lower side. This is how the position is. Now kidney, the ureter comes out. You can see these are the two tubes-like structure which comes out. Ureter. These are the ureters which are coming out. This is the urinary bladder and this is the urethra. Let's first talk about the kidneys. So if you look at the kidneys are the reddish and brownish in color, right? They are bean-shaped. Very important. They are bean-shaped. They are bean shaped. Next is length is around 10 to 12 centimeter, width is 5 to 7 centimeter, and the thickness is 2 to 3 centimeter. If you look at the kidneys, they are having two different parts. The outer part is termed as cortex and inner is termed as medulla. Their position is retroperitoneal. That means on one side the peritoneum is present. It is retroperitoneal in position. Now next structure is a ureter. We have a two ureter present coming from each of the kidney. One sac-like structure, urinary bladder, and one urethra is present. Clear? Now let's go deep into the topic of the kidney. All the diagram which you can see over here, they are directly from your NCERT, so that you should not get confused regarding the diagram. Now here you can see the section of a kidney. This is the LS of a kidney, longitudinal section of the kidney. Here if you'll see that there are two coverings present. The outermost covering is a renal fascia. Renal fascia is a connective tissue layer, right? It's a connective tissue layer. It's the function is the protection. It helps in protection. Now, the innermost layer is termed as renal capsule. This is also made up of the connective tissue, but the dense irregular connective tissue is present. In between them, the adipose tissue is present. So, adipose tissue act as, adipose tissue help as a shock absorber. So, it helps in absorption of the shock, right? So, renal fascia is present towards the outer side. This is present in the middle, right, and this is present in the inner side, towards the inner side. Yeah. Now if you look at the section over here you can see a region is present which is termed as cortex. So outermost part is termed as a cortex, and centermost part, this is termed as here, this is termed as medulla. Clear? Is this point done? Now you can see some triangular structures like this. These are termed as medullary pyramids. Because they are present in the medulla, so they are termed as medullary pyramids. Medullary pyramids are present. The functional unit of the kidney, what is that? The functional unit of the kidney is the urinary vestibule or the nephrons. Nephrons are present in the medullary pyramids. So how they are present, that I'll talk to you later on. These are the nephrons present over here. Now there are part of cortex which goes into, you can see this region, this region. There's a part of cortex which go deep into the medulla region. This is termed as renal column. Another name is renal column of Bertini. Renal column of Bertini. Renal column of Bertini is present. Clear? Renal column of Bertini. Now next region, you can see the medullary pyramids, they are present. Their number varies from 8 to 11. So their number varies from 8 to 11. Clear? Now the next one, all of these pyramids, they open into the calyx, right? This is termed as calyx. Calyx has two parts. The one which you can see over here, buddies, this is a major calyx. This is the major calyx. Later on it opens into the minor calyx. Two regions are there. So you can see all the medullary pyramids, they open into a region which is the major calyx, and then it opens into the minor calyx. 2 to 3 minor calyx region are present. Now later on it opens into this region here. Region is present which is termed as renal pelvis. From renal pelvis the urine comes out from the ureter. Now listen very, very, very carefully. I'll give you an overview of this topic once again. This is the kidney present. So kidney, medullary, see, urine is formed in the medullary pyramid. From medullary pyramid, urine will come into the major calyx. From major calyx to the minor calyx. Clear? Yes. Then into the pelvis. Then into the ureter. Clear? Done? Clear? Hope this is clear. Now then into the ureter, from ureter it will enter into the urinary bladder, and from urethra it will come out. Now there is a region of the kidney. Guys, can you see? This is a region of the kidney. Here, this is one region of the kidney. If you draw a structure of a kidney, how will you draw? You will be drawing it like this. Clear? Now this region here, this particular region, this is a concave region. Concave region. What is this? This is a convex region. Two different regions are present, convex and concave region. Now these two regions which are there, they are super important. Now, from the concave region, you will see a ureter is coming out. Not only the ureter, in fact, the arteries and veins. This is a place for the entry of the arteries. Arteries will come in. Here, the veins goes out. So, this is that region. Hilum. Another name is hilus present. Clear? Now, moving on to the next, you will see the next structure we are having is the nephron structure. Let's look at the detailed structure of the nephron. So, this is how a nephron is present. Guys, can you see? Clear? Clear structure? Now, if you look at the structure here, this is one. Clear? Here. Guys, can you see? Now, this is termed as the Bowman capsule. The one which I have drawn over here, this is termed as a Bowman capsule. Bowman capsule, right? Now, in the Bowman capsule, the afferent arteriole comes and efferent arteriole goes. The afferent arteriole come and efferent goes. So, question comes like, what is afferent arteriole? Afferent arteriole, afferent arteriole is that arteriole which is thick in diameter. It is thick in diameter. And you know from where it is coming? It is a part of the renal artery. So, a branch of the renal artery comes in the form of a renal arteriole over here. Here in the center, you will see the glomerulus is present. What is this? Glomerulus. So, glomerulus is what? Glomerulus is a bunch of capillaries. What is this? This is the capillaries present. Here you will see lots of capillaries are present. What are present? Lots of capillaries are present, which are actually not branching, in fact. I'll show you how these are present. Now, this is the afferent arteriole coming. Now, look at this. This is how it is present. Can you see? This is termed as the glomerulus. So, glomerulus increases the surface area for the filtration to occur. Now, here the Bowman capsules is present. And this Bowman capsule helps in the filtration. Here the filtration, ultrafiltration happens over here. Right? Ultrafiltration happens over here. Now, the next part you can see, I'm using the black color now for the color. Now, you can see here the coiled region. Guys, can you see the coiled region which I marked with the black color? What is this? This is termed as a proximal convoluted tubule, which I will be referring later on as PCT, proximal convoluted tubule. In the glomerulus, filtration occurs, right? Ultrafiltration occurs. And the PCT is that region where the 99% of the substances, they are being reabsorbed. The next region, which you can see, one second, this you can see, which is coming down and then it is going up. This region is termed as a loop of Henle. This region is termed as loop of Henle. Okay, let's do one thing. Let's make a diagram. One second. Okay. Now, let's make a diagram and let's try to understand the structure. I am making an extended form of this structure. Clear? This is a structure of a nephron. Here, as I have told you, here the afferent arteriole comes and efferent arteriole goes. Here there is a bunch of capillaries which is termed as glomerulus. Clear? Afferent arteriole and efferent arteriole. So what is this? This is the afferent arteriole. What is this? This is the efferent arteriole. Clear? This is termed as the Bowman's capsule. Bowman's capsule. What is the name of this reddish structure? This is termed as glomerulus. Together, Bowman capsule and glomerulus, they are termed as Malpighian corpuscles. Malpighian corpuscles. Clear? Done? Clear? Now, if you will see over here, now this is the Bowman capsule, and the next structure which you can see over here, what is this? This is the PCT, proximal convoluted tubule, right? Now this whole structure, guys, can you see from here till here? What's the name of this structure? The name of this structure is the loop of Henle. Loop of Henle. What is this? The loop of Henle. What is the name of this structure? This is the ascending limb. Sorry, this is the descending, because this is going down. So this is the descending limb. Right? What's the name of this? This is the ascending limb. Ascending limb of loop of Henle. Clear? Now here this region is the thick ascending limb. This is the thick ascending, because this is thick. So this is thick descending loop. This is the thin descending loop. This is a thin ascending loop. This is the thick ascending loop. Clear? Now this is termed as distal convoluted tubule, the DCT. This is termed as collecting duct. This is termed as collecting duct. Collecting duct is not a part of nephron. Please remember, this is not a part of nephron. So hope the structure is clear. Here in case of PCT and in case of DCT, very important. Here the brush bordered, I'll write it here. In case of PCT and DCT, the brush bordered cuboidal epithelium is present for the more reabsorption of substances. Clear? Is this point clear? Let's go deep into the structure here. Now you can see this is the structure of a Malpighian body. Another name of Malpighian body is the renal corpuscles. Here the afferent arteriole is coming and efferent is leaving. You can see the glomerulus is present. Then you can see the Bowman capsule is present. So here the lining, guys, can you see this lining? This lining. Here the special, here also buddies, the squamous epithelium is present. Which epithelium? The squamous, the flat cells. The squamous epithelium is present. Squamous epithelium. In this case, the special cells are present. Special cells, I'll make that with this color. Special cells are present, which helps in filtration, which are having villi-like structure, right? These are termed as podocytes. Podocytes. Podocytes forms this area. Guys, can you see? This. What is this? This is a filtration slit. What does it form? Filtration slits. So with the help of filtration slits, easy filtration can happen. Yeah. Now, what all things are present? Here you should know the endothelium lining of the capillaries will be present, which will help in the filtration. Now next one, you can see the squamous epithelium of the Bowman capsule will also be present, which helps in the filtration. More the thickness of membrane, more it will become difficult for the filtration to occur. Clear? From the next region that will start after here, that is a proximal convoluted tubule. So one which is present over here, that will be blood. Here what will you find? Blood. Blood coming over here. Here after the filtration, the substances will come over here. These substances will not be termed as urine. That will be termed as, here the substances which will be present, they will be termed as filtrate. Filtrate, right? Later on after DCT, in the collecting duct, it will be termed as the urine. Now, let's talk about the mechanism of urine formation. Now, let's understand this concept of the urine formation. That how does a urine formation occur? So, here 3 steps are involved. One is ultrafiltration. One is ultrafiltration. Second is the tubular reabsorption. And the third one is secretion. So I'll be explaining all these steps with you one by one, right? First, let's talk about ultrafiltration. Ultrafiltration. So because of ultrafiltration, what happens is we will get filtrate. Let's draw the diagram again so that I can make you understand the concept. Now, this is how the structure look like. Here the glomerulus, here the efferent arteriole, here the broader one is the afferent arteriole, guys, right? Now the blood will be present over here, and here the filtration will occur. Now let's talk about the composition of both of this. Here you will see here the total blood which comes over here, total blood which comes to the renal artery. What is that? That is around 650 almost ml, that is 1100 to 1200 ml per minute. This much blood comes in. Clear? Is this point clear? Now, out of the total blood, if I have to calculate only the plasma, because the blood cell will not be participating in the filtration. Blood cells, here the participation will not occur. So only the plasma is the one which will be filtered. So the plasma will be around 625 ml per minute coming in the afferent artery. Clear? Now just imagine blood is coming with the pressure. Now the diameter of the pipe is reduced. So there will be a pressure generation. Because of that pressure, there will be formation of the filtrate. So amount of filtrate which will be formed by this plasma is 125 ml per. So that means here 125 ml of the filtrate is formed per minute. Clear? Now out 125 ml of filtrate will not be involved in the urine. So here, if you look at the urine which is formed in one day, that is 1 to 1.5 liter per day. So there is involvement of the further reabsorption, which is the selective reabsorption. Selective substances reabsorption will occur. Clear? Now there are various you can say forces which work over here. But here I'm just discussing, because it's a revision session, so I'm just discussing the net filtration pressure. So what will be the net filtration pressure? So net filtration pressure, that is almost the net filtration pressure. So what is the net filtration pressure? The 10 mm of Hg. 10 to 20 mm of Hg is the net filtration pressure. Why? Because actually the glomerular filtration pressure, that is around 60 mm of Hg. Later on, there is a blood colloidal osmotic pressure. There will be hydrostatic osmotic pressure that will be working against it. So the net filtration pressure is 10 mm of Hg. Is this point clear? Clear? So how many filtrate is formed? 125 ml per minute. But how much we produce? We produce very less. So that means mostly they are being reabsorbed. Is this point clear? Now the next thing comes. What is the filtrate? So filtrate is equal to plasma, but here the proteins are not present. Major proteins. I am saying only the major proteins are not present. Minus major. This is the filtrate. Done. Let's move on to the next step. That is the selective reabsorption. Clear? Let's talk about the next step, which is the selective reabsorption. Step number 2. Reabsorption. Yeah. Or the reabsorption. Clear? Now here you will see that some arrows, they are coming out of the PCT. Can you see? Some arrows are coming out of the PCT. Bicarbonate, NaCl, water, nutrient, K plus ion. So that means what is happening? This is the reabsorption. This is the reabsorption. What is this happening? Reabsorption. So from the PCT, if you look at the PCT, 70 to 80% of the substances, they will be reabsorbed. That include water, NaCl, bicarbonate. That include your nutrient in the K plus ion. Now please remember here, in case of reabsorption, here, glucose and amino acids are completely reabsorbed. These are the points which you have to remember. Completely reabsorbed. These are the things which are completely reabsorbed. Glucose and amino acid completely reabsorbed. That means you will, in a normal condition, you will not find glucose or the amino acid in the urine. Please remember, and all thanks to the PCT. So you should remember, this is the things which occurs in the PCT. Now let's talk about more reabsorption. Where does the more reabsorption occur? You will see that deep down the medulla region. Now one question which comes from this topic, where is the PCT present? PCT is present towards the cortex region. In the medulla region, you will find the loop of Henle's are present. In the loop of Henle, there will be reabsorption of water. In the descending limb is only permeable to water. So only water is being reabsorbed from the descending limb of loop of Henle. Done, guys? Clear? Now, only water is being reabsorbed. Clear? Is it permeable to NaCl? I'll say no. It is not at all permeable to NaCl. Now, there is a loop formation. This is termed as hairpin loop. What do we call this? This is termed as hairpin loop. This is termed as hairpin loop. This is the ascending limb. From ascending, NaCl is coming. From another ascending, the NaCl is also coming. That means here, the reabsorption will be active. Here, the reabsorption will be passive reabsorption. Clear? NaCl, now it is impermeable to water. This is impermeable to water, whereas the one which is on this side, that is a descending limb, this is impermeable to salts. Now next region, this is termed as a distal convoluted tubule. Distal convoluted tubule is that region where the conditional reabsorption occur. What will occur? Conditional reabsorption. Conditional reabsorption. Conditional reabsorption. So what do I mean by conditional reabsorption? Conditional reabsorption means whenever there will be role of aldosterone hormone. Aldosterone. Here the reabsorption of the sodium ion, the NaCl, will occur. Clear? Whenever I'll be saying the role of ADH, antidiuretic hormone, here there will be reabsorption of the water and NaCl. Water and NaCl reabsorption. Conditional. That means something is coming and something is telling the nephron for the reabsorption. Point done? Clear? Now, hope this point is clear. Let's move on to the next part, which is a collecting duct. In the collecting duct also, the reabsorption occur. Here, there is a reabsorption of Here there is a reabsorption of the water, water and only water. Now next one is the secretion. Now what is a secretion? Please remember buddies, here there is secretion. That means from outside there will be secretion of the H plus, the NH3, H plus NH3 and some K plus creatinine also. Right? These are substances which body has to remove. So their secretion occur in PCT. Now, in case of DCT, there is K plus and H plus. And please remember, here, from collecting duct, some urea comes which is absorbed over here. That is in the ascending limb, right? So, yes, it plays a great role. Small amount of urea that is reabsorbed from the collecting duct, that is again taken up by the ascending limb of loop of Henle. So from ascending limb of loop of Henle, it will be circulating like this, and then small amount will come, and like this the circulation is important. Why it is important, that I'll tell you later on, because it has a great role in the counter current mechanism. So I'll be explaining you over there. Clear? So hope this point is clear, buddies. Now let's move on to the next topic, which is a counter current mechanism. Counter current mechanism, that means something counter is happening. Now, where is counter current mechanism existing? Counter current mechanism, that means if you look at this loop of Henle, they always runs in opposite direction. Both loop of Henle runs in opposite direction, so they form a counter current. Now the next region you will see here. What is this? This is termed as vasa recta. What is a vasa recta? Vasa recta is a part of efferent arteriole that also runs in opposite direction. So they are in counter current to each other. Now let's come to this diagram and let's understand here. From efferent arteriole, the blood vessel which is coming out from the nephron, efferent, will be dividing further. It will form a peritubular capillaries all around the nephron. Here, when it is around the distal convoluted tubule, when it is around the loop of Henle, they form a loop-like structure which is termed as the vasa recta. This is termed as vasa recta. So they form a counter current mechanism. So this counter current mechanism, this is important for maintenance of osmotic gradient. Osmotic gradient from cortex to medulla. Cortex to medulla. Clear? Okay. Clear? Now. Now, look at this diagram, buddies. Here, what is this? This is a descending limb of loop of Henle. So the blood which comes over here, the blood, whatever the blood comes, it is having a particular osmolarity. And you know what is that osmolarity? That osmolarity is around 300 milliosmol per liter. 300 milliosmol. Clear? Later on, from this region, water comes out. Water comes out. As the water will come out, the osmotic gradient will increase from 300 to 400, from 400 to 600, from 600 to ultimately 1200 here. Point clear? Is this point clear? Hope you're able to understand this topic. Clear? Now, 1200. This is 1200. Now, from this region, from ascending limb of loop of Henle, the NaCl comes out. That means from 1200, it will become 900, 600, 400 and 300, and ultimately here it will become 200. It will become here 200. Clear? Now, this increase in osmolarity and there is a decrease in osmolarity. From here the water comes out, from here the NaCl comes out. Clear? Clear? This proximity is clear. So from one region water is coming out. When water is coming out, the osmolarity is increasing. When NaCl is coming out, the osmolarity is decreasing. Clear? This point is done. Done. Clear? Now moving on to the next part. So blood which enters in the vasa recta, it is having the osmolarity of around 300 milliosmol per liter. What is osmolarity? Here also the 300 milliosmol is present. How much? 300 milliosmol. Now this 300 milliosmol osmolarity, the particular osmolarity which is present, here what happens is the NaCl which is coming out from the ascending limb of loop of Henle, this is being taken up by here. That is the descending limb of vasa recta. So what will happen? The osmolarity will increase. So ultimately it will become 1200 osmolarity. But we have to maintain a particular osmolarity, particular osmotic concentration of the blood. Now in this case, as the blood moves towards the upper side. As the blood moves towards the upper side, you can see over here, as the blood will move towards the upper side, the NaCl comes out. It has to throw NaCl towards the outside. NaCl towards the outside. Clear? So ultimately the blood which comes out will also be 300 milliosmol per liter. Is this point done? Clear? I'll explain you this once more. This is right. This is the structure, right? What is this? What is this, guys? This is the loop of Henle, descending limb of loop of Henle. What is this? This is the ascending limb of loop of Henle. Clear? You will see the peritubular capillaries are present over here, and these peritubular capillaries, this is what? This is the descending. What is this? Descending vasa recta. Here, this is the ascending vasa recta. Vasa recta. Clear? This point clear? Very simple. And now I'm going to make this topic super duper easy for you. Now, you know the filtrate is coming over here. You know what's the osmolarity of filtrate? 300 milliosmol. Because in PCT, water is also reabsorbed, the salts are also reabsorbed. The osmolarity, it becomes 300 only. Now, as it goes down, the water comes out. Water comes out. This is the water which is coming out. Water coming out, right? You know, descending limb is permeable to water only. So the osmolarity will increase to 600, then again to 900. Here it will become 1200. Clear? Very easy peasy lemon squeezy. Now moving on to the next, here the NaCl will come out. NaCl comes out. NaCl comes out. So what will happen? The osmolarity will further decrease. What will the osmolarity? Here it will be 900. Then it will decrease to almost 600 here. It will decrease to 300. And ultimately there is so much of reabsorption that in DCT it becomes 200 milliosmol per liter. Done. Clear? The descending limb of, the vasa recta is also in proximity with the loop of Henle. But here the flow of blood is opposite to that of flow of filtrate. If filtrate is flowing in this direction, the blood will flow in the opposite direction, that is from this side to this side. So from here, you have to listen to this point very carefully. Here what happens is, blood is coming with the osmolarity of 300 milliosmol per liter. Blood always maintains the osmolarity to 300 milliosmol per liter because we cannot afford beyond it. Clear? Now, here what will happen? The NaCl is reabsorbed. As the NaCl is reabsorbed, it will come out to be 600, then to the 900, and ultimately to the 1200. It will reach to the 1200. Is this point clear? So from the 1200, here again blood wants that whatever the blood leaves, it should have the same osmolarity, that is the 300 milliosmol per liter. In that respect, from this region, NaCl comes out. NaCl comes out. So here the osmolarity becomes 600, sorry, 900, then 600, and ultimately the 1200, 300. Clear? This is a countercurrent mechanism. Similarly, in the interstitial fluid also, outside also, the same osmolarity is present because of the NaCl, right? You know the NaCl is coming out from here, so NaCl will settle over here. As it will settle over here, it will become 1200. It will be 1200. Similarly, the urea is also coming. Urea will be absorbed over here. Small amount of urea in the ascending limb of loop of Henle. That is important for the maintenance of high osmolarity in the medullary region. That is the reason urea is reabsorbed. Now you know the importance of urea. Now let's jump to the another topic, that is a regulation of the kidney function. Main topic, which is the regulation of the kidney function. First, let's talk about the renin-angiotensin aldosterone system. Renin-angiotensin aldosterone system. Here, what do you need to remember? So guys, you have to remember whenever blood osmolarity decreases, or I am saying the blood pressure decreases, the glomerular filtration rate GFR decreases. Yeah? This will evoke the JG cells, juxtaglomerular cells, special cells present in the Malpighian body. JG cell will secrete the renin, a hormone it will release which is termed as renin. Now this renin will be responsible for the conversion of angiotensinogen into angiotensin 1. Angiotensin 1, right? It will help in this conversion. This is coming from the liver. Right? Angiotensin 1 will be converted into angiotensin 2. And here the angiotensin converting enzyme which is coming from the lungs is important. Now, this angiotensin 2 will be having following role. First, it will go to the adrenal cortex. And adrenal cortex will secrete a hormone which is aldosterone. Right? It will, so aldosterone, you know what's the role of aldosterone? It helps in reabsorption of sodium and water from DCT. NaCl and water reabsorption from DCT. Here. Now the next thing is it will act as a vasoconstrictor. Due to which blood pressure increases. This will be responsible for increasing the blood salt concentration. So blood volume increases, ultimately the blood pressure will also increase by the vasoconstrictor. Yeah, renin, angiotensin, aldosterone system. Next one we are having is the role of ADH. ADH. What is the ADH? ADH is antidiuretic hormone. So here also, whenever the BP reduces, glomerular filtration rate reduces. In that respect, posterior pituitary, which part? The posterior pituitary will release one hormone which is the ADH. Another name is vasopressin. Vasopressin. This will allow the reabsorption of water from DCT and CD. CD is collecting duct. Second is vasopressin. That means it will act as a vasoconstrictor. It will increase the BP, blood pressure. So ultimately, more reabsorption of the water as the BP increases, it decreases, and this occurs. Now, third one is the role of ANF. Atrial natriuretic factor. Atrial natriuretic factor. Whenever BP is high, blood pressure is high, the signal will go to the atria, heart atria. This is the heart atria. Atrial wall will secrete the ANF, atrial natriuretic factor. Here, what will occur? Here, this will increase the, this will decrease the BP. How does it occur? It will suppress aldosterone. It will suppress the aldosterone. Here, it will suppress the RAS, right? Basically, the RAS. Then, now, moving on to the next topic, that is a micturition. So, what is a micturition? That is expulsion of urine. So, here you all know we are having a central nervous system. We are having a central nervous system. This is how the urinary bladder is present. Here special type of muscles are present which are termed as detrusor muscle. Which muscle? Detrusor muscle is present. Here some stretch receptors are present. Stretch receptor. As soon as they will detect that there is increase in the urine, that is more than 500 ml, signal will go from here to the central nervous system with the help of afferent neuron. Afferent neuron. CNS will send the signal here and signal will send over here. Clear? This will cause here, this will cause contraction of detrusor muscle, smooth muscle. These are the smooth muscle, the contraction of urinary bladder, and this will cause relaxation of internal sphincter. Internal sphincter. This is the internal sphincter present. Internal sphincter. This is the outer sphincter present. Outside sphincter. This is involuntary. What is this? This is involuntary. And what is this? This is voluntary. right? So pressure will be feel, or the urine pressure will feel towards the outside. We'll go to the washroom and we will expel the urine. In many cases, yes, some babies, some kids, even when they are in 13 and 14 year of age, they are not able to control the urine. So that is because of the not proper training of the cerebral cortex. That region is the one which is responsible. Otherwise, it is the central nervous. Clear? Hope this point are clear. So these were some major topics which were there in this chapter. So see you guys in the next class. There are some other topics also, like I'll tell you. There are role of other organ. For example, the intestine also helps in removal of the nitrogenous waste, for example, the ammonia. Other thing is skin also helps in the removal of the substances from the body. For example, we are having mucous membrane also, which sweat glands are also the one which helps in the removal of the substances. So that is also a kind of excretion. Some disorders are also there. Very easy those disorders are. Please go through them. They are important for your NEET examination. But some major topics I have explained to you in this topic. How did you find the lecture? Do let me know in the comment section. Bye bye. Have a nice day. Thank you so much.